Genetics and inheritance, which is the third topic that you should be studying for paper 2 of your NSC final examination, has a large volume of work that you generally need to work through. Therefore, in this video, we will be going through a few questions taken from section B that focuses on some of the key concepts which you are required to understand and grasp in this topic. Some of the key points that you should focus on when studying genetics and inheritance. Firstly, complete dominance, incomplete dominance, and codominance. As stated in your exam guidelines, please consult it. The law of dominance, please consult the exam guidelines. Monohybrid crossing, understand and use the template, position of meiosis and fertilization, as well as your P1 and F1 on the template. Answer the question at the end, which is usually the question where you need to draw a genetic cross. Proportion and ratio of genotypes and phenotypes, stating the principle of segregation. Inheritance of sex, differentiate between sex chromosomes, which are your gonosomes and your autosomes. Sex-linked characteristics and disorders, the correct way of writing it, also in pedigree diagrams. Blood groups, the difference between genotype and phenotype of each blood group. Remember, there are different types of blood groups, namely A, B, blood group AB, as well as blood type O. The correct way of writing the genotypes. Then dihybrid crossing, the correct way of writing it. If you have the genotype, for example, 2 capital G's or 2 capital R's, or a capital G, a small g, a capital R, a small r, how to write out your gametes for a dihybrid cross. Distinguish between dominant phenotype and dominant allele. Mendel's principle of independent assortment. Then pedigree diagrams, interpretation of pedigree diagrams. Give the phenotype and genotype. State which allele is inherited from each parent. The examiner does not have to provide a key. Please remember that. In mutations, the different types of mutations, that's your gene and chromosomal mutations. Different effects of mutations, harmful, useful, and harmless mutations. Refer to the exam guidelines to consult this as well. Genetic engineering, you had stem cells, genetic modification and cloning, sources and uses, benefits, the advantages and disadvantages, a brief outline of the processes. Then paternity testing and DNA profiling, describe how paternity testing is done, not only identifying the father in a diagram, but describe how the mother, father, and child's DNA bars play a role. The first question that we will be looking at is taken from question 3 from 3.1 that states Menkes syndrome is caused by a sex-linked disease. It's caused by a mutation in the gene coding for a protein called ATP7A, which is responsible for the transporting of copper into cells. Symptoms include easily broken hair, a loss of early developmental skills, weak muscle tone, sagging facial features, seizures, and nervous system deterioration. This is the pedigree diagram that has been given for this question. You have keys for the normal female, for the normal male, and the male with Menkes syndrome. The first question, 3.1.1, says name the type of diagram shown above. So what is the name of this diagram? It's going to be a pedigree diagram. 3.1.2, define a sex-linked disease. So what do they mean by a sex-linked disease? A sex-linked disease can also be referred to as a mutation, a gene for disease found on the X or the sex chromosomes or found on the gonosome. So it's a gene for disease found on the sex chromosome or on the gonosome. 3.1.3 says how many generations are shown in the diagram. Remember that each row represents a generation and you have 4 different rows. Therefore you have 4 generations. 3.1.4 is Menkes syndrome recessive or dominant? Now if we're looking at Menkes syndrome, we know that it's going to be a sex-linked genetic disorder, and we're trying to figure out if it's going to be recessive or if it's dominant. In order for us to figure that out, we need to look at what is actually happening on the pedigree diagram. If we look at our first set of parents, we can see that none of them were affected. but they had 1, 2, 3 children and one child was affected. So that then means that that child inherited a allele from either both parents or from one of the parents. If both parents were unaffected and only one child is affected, then it's only obvious that the Menkes syndrome is going to be recessive and not dominant. So 3.1.5, give a reason for your answer in question 3.1.4. We can see that individual number 5 is affected. But if we looked at individual number 2 was not affected. Then if we move further down, we go to individual 15. We can see individual 15 is affected, but both of the parents were also unaffected. Individual 10, which is the mother, was also unaffected. If we go to individual 13 was affected, both the parents were unaffected, including the mother was unaffected as well. So to answer 3.1.5, first individuals 2, 7 and 10 do not have Menkes syndrome. 2, 7 and 10 do not have Menkes syndrome. However, they carry the allele. So they will carry the alleles because they have a child 5, 13 and 15. So 5, 13 and 15, the children has Menkes syndrome, and therefore we are going to say it is going to be recessive. 3.1.6, use symbols XR, X more R to provide the genotypes of the following individuals. Individuals 2. So if we go to individuals 2, we already said that individual 2 is unaffected, but it is going to carry that recessive allele, and it's a female. Therefore we need to have 2X, and one is going to have the dominant allele and the other one has the recessive allele. Then individual 5, we said that individual 5 was affected, but it was a male that was affected. Therefore a male has an X and Y, but on the X you are going to carry that recessive allele that's going to give the male Menkes syndrome. 3.1.7, you are using a genetic cross to show the chances of the next child born from individual 13 and 14 who has X with 2 capital letter R's will be born with Menkes syndrome for 6 marks. So chances of the next child born from individual 13 and 14, so we can see they gave us the genotype for individual 14. We need to come up with our own genotype for individual 13, but we know that individual 13 is a male with Menkes syndrome. So we start off with 3.1.7, P1, our phenotype. It's a male with Menkes syndrome and it's a normal female. The genotype of the male is going to be X with small r and a Y because the male is affected. But the normal female, we got the genotype from the pedigree diagram. We then moved on to meiosis and we got our gametes. After the gametes, we went on to the process of fertilization where we do the Punnett square. We got our offspring for the Punnett square. Therefore, in our F1 generation, we had 2 normal females and 2 normal males. So the percentage chance of the next child having Menkes syndrome is therefore 0%, because you had 2 normal males as well as 2 normal females. 3.1.8, explain why it is more common for boys to suffer from Menkes syndrome compared to girls. Very easy question. Please make sure you understand this. It's an easy way to score marks. So because Menkes syndrome is going to be a 6-linked genetic disorder, it's going to be carried on the X chromosome, but boys, males, generally only have 1 X chromosome, so they don't have that additional X chromosome to mask that recessive allele as females do. So males only have 1 X chromosome. If they carry Menkes syndrome or the allele for Menkes syndrome on the X chromosome, then they have the disease. They don't have a second X chromosome to mask the recessive alleles as females generally do. The next question is 2.3 that says Duchenne muscular dystrophy is a neuromuscular disorder and is caused by a recessive allele X with a small d on the X chromosome. The allele for normal neuromuscular functioning is X with a capital letter D. Therefore, to be affected, it's going to be carried on a recessive allele. The inheritance of DMD in a family is shown in the diagram below. So you have a pedigree diagram. How many generations are shown in the family? You have 1, 2, 3 generations. Therefore, the answer is 3. 2.3.4 explain why individual A can only be heterozygous for this condition for 3 marks. So we're going to individual A. Why can individual A only be heterozygous for 3 marks? So first we can see that individual A is unaffected. Individual A is unaffected. If we go to the key here, it's unaffected female, caused by a dominant allele on the X chromosome, the X with capital letter D. So to be unaffected, this needed to be an X with a capital letter D. The offspring C inherits Y chromosomes from individual B or the father. So offspring C inherits that Y chromosome from the father, which is individual B. And it inherits the X or the small letter d from individual A. So because individual C is affected and it's an affected male, it must have inherited that X or the small letter d from the mother. Therefore, individual A is going to have X with the capital letter D and X with a small letter d, which brings us to the conclusion that individual A must be heterozygous. So answering 2.3.4, individual A is unaffected, caused by the dominant allele on the X chromosome. Offspring C inherits the Y chromosome from individual B or the father, but it inherits the X chromosome with a small letter d from individual A, which is the mother, and therefore individual A must be heterozygous. So question 2.3.5 says individual E marries a woman who is normal for neuromuscular functioning but carries the recessive allele. Use a genetic cross to show the percentage of offspring that will have DMD. So for 2.3.5, we can say we see the phenotype is a normal male. For the female, it's a normal female. That is the male's genotype, the female's genotype. We then draw our Punnett square after we have found our gametes. We fill in the Punnett square. We see that in our F1 generation, our genotypes, our phenotypes, we have 3 normal unaffected, 3 normal that was unaffected, and 1 with DMD that was affected. Therefore, there's 25% chance of the offspring having DMD because only 1 out of 4 was affected. So 1 out of 4 multiplied by 100 gives us 25% chance. The next question, 2.5, says in tomato plants, the allele for tall plants is dominant over the allele for short plants. And the allele for red fruit is dominant over the allele for yellow fruit. A red fruit tomato plant that is tall crossed with a yellow fruit short plant. The Punnett diagram below shows the possible gametes produced by each parent. So you have the possible gametes produced by each parent. Remember, you could use these gametes to also find out what the actual genotypes of each parent would be. So if we have to go to the first one, we could say that their genotype is going to be capital letter T, small letter t, capital letter R, small letter r. If we go to the second parent, we can say their genotype is 2 small letter t's as well as 2 small letter r's. 2.5.1 state the type of cross represented in the diagram. There are 2 types of genetic crosses. It's either the monohybrid cross or the dihybrid cross. Because we are looking at 2 characteristics here, we are looking at the height as well as the color. Therefore, it's going to be a dihybrid cross. Give a reason for your answer. It involves the inheritance of 2 characteristics because we are looking at the plant's height as well as the color of the fruit. 2.5.2 give the phenotype of offspring X. So we are looking at offspring X. We can first look at the genotype. So we'll have capital letter T, small letter t, small letter r, but I want the phenotype. Capital letter T is masking the small letter t, therefore it's going to be tall, and 2 small letter r's is going to mean it's going to be yellow. So therefore it's going to be tall with yellow fruit. The genotype of offspring Y, so we are going to give the genotype of offspring Y. We can simply use it according to what's given here. So small letter t with small letter t, and capital R with small r. Therefore, that is going to be the answer. Two small letter T's, a capital R, and a small r. That is simply going to be the genotype. 2.5.3. What percentage of offspring is expected to be recessive for both characteristics for 1 mark? What percentage of offspring is expected to be recessive for both characteristics? Now, if you look at the last column here, you can see that you have that both those traits represented in the gametes is recessive, and here they are all recessive as well. Therefore, everything going down here is going to be complying to the answer in 2.5.3. You're going to have small letter T's, small letter R's going all the way down. And how many blocks are there? 1, 2, 3, 4. And how many blocks are here in total? There are 16 blocks here in total. So you're going to have 4 over 16 multiplied by 100 would give you your 25% as an answer. The next question, 3.4. Galactosemia is caused by an autosomal recessive allele with a small letter G. People with galactosemia are unable to break down galactose from dairy products like milk and cheese into glucose. So you have a pedigree diagram along with keys represented at the bottom. The first question, 3.4.1, says write down the phenotype of Ngizwe for 1 mark. So we go to Ngizwe. We can see it's a block that is unshaded. Phenotype, we simply give the characteristic an unaffected male. All the possible genotypes of Kelly for 2 marks. So we go to Kelly, genotypes. Remember, we are giving the letters. So Kelly, it's a circle. So circle, it's going to be a female that's unaffected. If the female is unaffected, remember this is an autosomal recessive allele that is going to cause galactosemia, but she's unaffected, she does not have it. Therefore, she can't have a small letter G, she needs to have capital letter G. So either she's going to have 2 capital letter G's or she can have a capital G and a small g, because she can either be homozygous dominant or she can be heterozygous, and it still means that she's going to be unaffected. Therefore, the answer is going to be capital letter G, small letter g, as well as 2 capital letter G's. That is all the possible genotypes of Kelly. 2.4.2, explain why stomata is definitely heterozygous for 3 marks. So we go to stomata. Why are we saying this one is definitely heterozygous? We got to trace it back to the parents. We see both of the parents were unaffected. So if both of the parents are unaffected and stomata is also unaffected, if we look at stomata's children, 2 of the children are affected. But Tyler is also unaffected. That means that the children inherited 1 recessive allele from each parent. Therefore, for 3.4.2, stomata is unaffected and has Zandile and Sonwabili who are affected. Therefore, they must have inherited a recessive allele, the small letter g, from each parent. 2.4.3, Sonwabili marries an unaffected woman whose mother had galactosemia. Do a genetic cross to determine the percentage chance of them having a child who has galactosemia. So Sonwabili, we go to Sonwabili, is going to be an affected male. An affected male means what? He is going to be affected for this particular genetic disorder. So Sonwabili marries an unaffected woman. If the male is going to be affected, because it's carried on to, it is autosomal recessive, it means he needs to have 2 small letter G's, but he marries an unaffected woman whose mother had galactosemia. So that means that this woman is going to be heterozygous. So you're going to take capital G and a small g. You'll first start with your P1. It's going to be, you'll state the phenotype. It's an affected male with an unaffected woman. The phenotype, you're going to have affected and unaffected. Genotype, 2 small letter G's for the male. Heterozygous for the woman. After meiosis, you're going to have your gametes. You then draw your Punnett square and complete the Punnett square. After that, you're going to get your F1 generation. After you underwent fertilization, In the F1 generation, you will notice that you're going to have 2 affected and 2 unaffected. So 2 are affected and 2 are unaffected. Therefore, there's a 50% chance that the child is going to have galactosemia because 2 of them are going to be affected. So 2 out of 4 times 100 is going to give you your 50%. The next question, 2.5, says BT corn is a crop that has been genetically modified to be insect resistant. The table below shows the percentage of land used to grow BT corn and the amount of insecticide used in a certain country between 1995 and 2010. So you have the years, you have the land used to grow BT corn in percentage and the amount of insecticide used per kg per hectare. 2.5.1, describe how genetic engineering of BT corn was done. Now, what do we do in the process of genetic engineering? The gene for insect resistance is isolated from another species and inserted into the DNA of BT corn where it is expressed. So that is how genetic engineering would have taken place. The gene for insect resistance, because we said the BT crop has been genetically modified to be insect resistant. So we took out the gene for insect resistance, isolated it from another species, inserted it into the DNA of corn where it was expressed. 2.5.2, describe the relationship between the land used to grow BT corn and the amount of insecticide used. So here you have the land, you have the amount of insecticide. You see the percentages of land is increasing, but the amount of insecticide is going to be decreasing. So therefore 2.5.2, as the percentage of land used to grow BT corn increases, the amount of insecticide used decreases. 2.5.3, state 2 other plant characteristics that are genetically engineered to increase food security. So what else do we do to plants to genetically engineer them to increase food security? We give them a longer shelf life, we make them drought resistant, we allow them to have more nutrients, frost resistant, increased size, increased crop yield, herbicide resistant, no allergens, heat tolerant, disease resistant. So these are some of the things which you could have stated any 2 of these for 2.5.3. 2.5.4, you are drawing a bar graph to represent the percentage of land used to grow BT corn from 1995 to 2010 for 6 marks. So you are using this table to draw your graph. You have your heading where you include both your independent and dependent variable. You place the independent variable on the x-axis, your dependent variable on your y-axis. You plot your bars, you have a correct scale. Therefore, you have 1 mark for drawing a bar graph, 1 mark for your caption, 1 mark for labeling your x and y axis correctly, 1 mark for having the correct scale on the y axis, and 2 marks if you've plotted your points correctly. The next question, 3.1, states in a certain country, 25,000 individuals were suffering from hemophilia between 2012 and 2020. Hemophilia is a genetic disorder that's caused by a mutation. A pie chart below shows the percentage of individuals with different levels of hemophilia in the country. So you have the pie chart, portion of it is going to be low with 35, a portion high with 40%, and a moderate portion with 25%. The first question, 3.1.1, says explain the effect of this mutation on an individual. So if you are going to have hemophilia, then what is the effect of hemophilia going to be? This is something that you would have studied in your notes. So hemophilia is generally going to prevent clotting of blood. And when it's going to prevent clotting of blood, it's going to be harmful. Why will it be harmful? Because it leads to excessive bleeding, internal bleeding, swelling of joints, and bruising. That is one of the effects that hemophilia is going to have. 3.1.2, calculate the number of individuals who were moderately affected by hemophilia in this country. Show all working for 2 marks. So we go to moderate, comes to 25%. But how many individuals were there? 25,000. Therefore, 25 over 100 multiplied by 25,000 gives us 6,250 after we type it into the calculator. 3.1.3 explain why it would be expected that most of the individuals affected by hemophilia will be males for 3 marks. Easy marks for this question. We know that hemophilia is going to be a sex-linked genetic disorder. Males are generally going to only have one X chromosome. It does not have that second X chromosome to mask the recessive alleles if the male is going to have hemophilia. Therefore, for 3.1.3, males have only one X chromosome, or they have an X and Y. If this chromosome carries the recessive allele, the male will have hemophilia, as there is no other allele that could mask the effect of the recessive allele. Males are not like females who have two X chromosomes, where that second X chromosome could possibly mask the recessive allele, and therefore it cannot be expressed in the phenotype. That brings us to the end of this video where we revise genetic and inheritance using section B questions from previous exam papers. Please ensure that when you are studying for paper 2, you are studying DNA, the code of life, meiosis, genetics and inheritance, as well as evolution. In the next video, we will be revising evolution using section B questions. Thank you very much for taking the time to watch this video. I hope to have you all join me in the next video, which will be revising evolution.